Hey guys, so we're going to start our notes part 3 and then I'll have part 4 for you guys next week. So let's take a look at part 3 of the notes. It's going to be on human heredity, pedigrees and karyotypes and all that. So let's go ahead and start your notes. Make sure you print out your student version to write on it or type directly on it, and then take pictures if you do that and upload that to Google Classroom. Okay, just like we've been doing. Alright, so human heredity. This is, there's two things you need to know. You need to know about a genome, which is your full set of genetic information that an organism carries in its DNA, and then also a karyotype, which is the picture showing the complete diploid set of chromosomes. And we'll look at an example of what a karyotype is here in a moment, right here. Here's your karyotype. It has two homologous pairs for each chromosome. And this is a typical human one. We have 46 chromosomes total, 23 pairs. This one has the, it's a male karyotype for this one. You have the X chromosome here and the Y chromosome here. That always comes at the end. We'll talk about that in a second. All right, transmission of human traits. And patterns of inheritance. So there's this thing called X chromosome inactivation. It only occurs in females where one of the X chromosomes is randomly switched off, and that will form this structure that you can see on a karyotype of a bar body. An example of an organism or a consequence of it would be calico cats. Their fur pigmentation color is actually X-linked. Next, we have human pedigrees. This is very similar to a family tree, but it is useful in gathering background genetic information and can be used for medical reasons. We are going to watch a clip of the Alzheimer's family that didn't realize they were all family until a nurse put a pedigree together of all of these patients and figured out that they were actually all some distantly related. Here's what a pedigree will look like. You need to write down what each part represents on your papers. So you guys have a black and white pedigree that looks just like this, almost similar. Make sure you put that a circle equals female and a square equals male. You have a horizontal line between these two, and that indicates that they are married. You have a vertical line down, which represents offspring. Okay? And the next thing you need to know is the sibling horizontal line goes in between like that, which would be different than this one here. The sibling one is up above, and the marriage one is in between. And then you have their offspring. Then circles are males or females, squares are males, and then shaded in means that person is affected by whatever we're looking at. In this case, we're looking at white forelock or no white forelock being present, and so if a person is not affected, it will not be shaded in, okay? So make sure you copy that down, pause it if you need to. Some disorders caused by individual genes. So we're going to look at several. The first is cystic fibrosis, which is abbreviated with CF. It is a deletion of 3 bases. So remember those A, T, G, G's, and C's. So just 3 gets deleted, and then you have this disease, and it produces a defective chloride ion transport protein, which probably doesn't mean much to most of you, but it basically causes serious digestive and respiratory issues. It's a recessive trait, so you would have to have it from both mom and dad in order to be expressed with it. And affected individuals with cystic fibrosis typically do not live into adulthood. The next disease we're going to look at, or I guess we're still going to look at a picture of this. Alright, so it's, if you want to take a peek at this and read through it, so you're literally missing 1 amino acid sequence. Boom. On chromosome number 7. The next disorder is Huntington's disease, which we are going to abbreviate with HD. This one is a dominant allele located on an autosome, which is not your sex chromosomes. It's the other 22 pairs. Pairs 1 through 22. This affects the nervous system. It typically has an onset in a person's 30s or 40s, so you don't typically know you have it until you're older. So when you're younger, you're fine. You can know that if you've got a family history of it, though, and can expect to see it at some point, maybe. All right, so now we're going to look at studying the human genome, and there are 3 steps to this. The first is cutting DNA. And when we do that, so you take your long strand of DNA, it has to be cut into smaller segments to actually study it. We use restriction enzymes to cut the DNA molecule into restriction fragments. Once we've done that, we then can use gel electrophoresis to separate it out and analyze those little fragments, and that's the separating DNA step. And then, after we've separated, then we can read it using DNA polymerase and dye-labeled nucleotides. We can then sequence that DNA and find out what is missing, if we're looking for something that's missing, or what sequence is actually there. All right, so the Human Genome Project was completed in April of 2003. It is designed to determine the nucleotide sequence of the entire human genome. We have figured out from this that there is about 21,000 genes. That's about 2 billion nucleotide base pairings. So that's one column is 2 billion down, and the other one would be 2 billion down too, because they're paired. So they revised it in August of 2018, and they have said that the number of genes only accounts for protein coding genes. So there's a whole hunk of stuff that we still have not interpreted yet, from what I understand. Okay, sharing data. So let's look at this as this pie chart, or not pie chart, Venn diagram. You're going to want to copy this down onto your blank one that's on your paper. Okay, so you can see my example one here. But we have 2 different things that kind of merge together. You have bioinformatics, which is your field of study combining molecular biology with information science. And then we have genomics, which is the study of whole genomes, including genes and their functions. And then we can combine those, and we use the data from both to get data analysis. We can make our visualizations, which would be models. We can make predictions from that. Okay. All right. Inherited diseases. Inherited diseases are genetic mistakes or mutations that are passed on to offspring. A carrier of an inherited disease is an organism that doesn't show the disease but passes it on. So they would have the recessive allele present, but they would not be showing it. Okay? So for example, we have sickle cell. And that is recessive, and it occurs when you have normal red blood cells that become sickle-shaped and clog blood vessels. But there's a reason that this has persisted, that we still have this, and that people who have it don't just die. That is because people who have sickle cell are actually adapted to survive against malaria. So that means it was beneficial, and that's why we still have it. Malaria is now treatable, but that's only been in the last, I don't know how many years, 50, 60, 100 years. Something like that. So genetically speaking, we wanted to have, or our bodies developed a way to combat malaria. Okay. All right. Sex-linked diseases. Disorders linked to X chromosome, sex-linked diseases are diseases or disorders that are linked to the X chromosome, which is your female chromosome. An example would be color blindness, meaning that you're blind to some certain colors, usually green and red, or shades thereof. The genotype for this, if you are a carrier, you would have, and this is XX is female and XY is male. So a carrier would have one normal X chromosome and then one X chromosome that has the affected gene on it. If you are a colorblind female, you would have to have both X's with it, with that carrier, or with the disorder on it, okay? If you're male and you don't have the carrier X, then you are normal. But here's where this is unfortunately why males catch it or have it more frequently, is because they only have one X chromosome. So if their X chromosome has it, they will be colorblind. They'll be affected by it, okay? So what we're going to do is we have a Punnett square on our paper. We are going to complete it, and we are going to cross a colorblind male with a carrier female. And I'm going to put it on paper for you, because I think that's the easiest way for us to do it. And let's get a fresh sheet of paper. I'm going to switch this over to our document camera. But let me write down our P1 cross. If we have a colorblind male, he's going to be XCY with a carrier female, so XXC. And I'll show you what that looks like here in a second. If we go down here, gotta get this on. Okay. So our affected male, colorblind male, has the X chromosome crossed with, I know it's hard with all these X's to see, a carrier female. So our male is going to go on top here because he was the first parent listed. And then our female is going to go on the side here. And these C's are in the subscript. So yes, last week we were working with the superscript with our blood types. This time we're working with the subscripts, so that it's going to be down below. All right, so this is the recessive trait. So that one is going to go on the right of our box. Okay, Y comes after X, so that one's going to be there. Okay, on the right. Then we're going to slide our X's across. And this one here is another carrier one, so make sure you put the C. Okay? Alright, so that we've completed our Punnett square. I don't think I asked anything else on the notes on that. I gotta see. Nope, just the P1 cross. Okay, so that's all we needed. Perfect. So hopefully you got that. Okay. Then we are going to, or I guess there's a little bit more information. Since traits are linked to the X chromosome, that means the male only needs to receive one X chromosome carrying the trait to have the disorder. He only has one X chromosome, so if it's recessive, you would have to have two if you were female to have the trait. So the female must have both X's with the trait to express the trait. Females with one sex-linked trait are known as carriers because that trait is recessive. So their normal X will dominate over the traded X chromosome. Okay. Non-disjunction in humans. Where did the... Nope, I missed a slide, guys. Where did it go? On our paper, we have the hemophilia one, so that must be one that we have to do on our paper instead. Let's do that. Sorry. No slide for it. So, that's correct. There's no slide for it. Okay, so we're going to look at this here. We have another trait, which is hemophilia, okay? And we're going to have our XN mean that that is an affected X, and X is unaffected without the subscript. Okay, so we are going to cross a female carrier. So our P1 cross should be, a female carrier would be XX subscript N with a normal male, XY. Because remember, carriers do not express the gene. So let's look at this and see what we've got. So let's make, so I've done our P1 cross. We have XXN for our carrier female and our normal Y. Let's make our Punnett square, shall we? So female is on top this time, because that was listed first. And then our XY is here. So X will go in front on the right. XN goes on the left here, or on the right here, sorry, left here on the right here, but on the right, left on this one, because Y's are going to go after. So X, X, Y, Y. Fill it in. There we go. And we need to get the number 1 is the number of offspring with hemophilia. So how many offspring here are going to be affected by this disorder? If we look at it. And make sure get this so that you guys can see everything. There we go. Okay, so the number affected is just this one. 1 offspring. Okay? The number of offspring without hemophilia. No hemophilia is actually going to be 3. All the rest of them do not have it. How many carriers do we have? That means they have it, but they're not expressing it. We have 1 carrier. And the number of males with hemophilia? And that's going to be just this one here. And number of females? With hemophilia is 0. No females have it. We have 1 carrier, that's why. Okay? Alright, so now we can go to non-disjunction. Okay, so what we're going to do for this one is we're going to actually copy some stuff down in the circles, and I'll show you in just a second. So when chromosomes don't separate, leaving 1 more chromosome in one gamete and 1 less chromosome in another gamete, that's when you have non-disjunction occurring. So this is part of meiosis where they would separate, and then we would have our alleles come off, like we would put the gametes on the outside of our Punnett square. So if you looked at this here, these are our gametes. Well, sometimes you are having them not separate out into the 2 different cells. So what we're going to write on our paper is you've got normal, okay, is right here. You have an X chromosome, or a pair of chromosomes, and then each half of it gets into each of the 2 gametes. But when you have non-disjunction, so you have 4 alleles, so you're going to draw them like how I would draw them, is like that, okay? And then 1 half goes here for this one, but then the other half goes here like it's supposed to, but this one doesn't separate. So you put both halves down here. Instead of being 2 in each, we have 1 and 3. Alright, hopefully that makes sense. Alright, so we're going to look at what happens when we have non-disjunction occurring. So I'm sure many of you have heard of Down syndrome before. This is another disorder we're going to talk about. And Down syndrome is when you have an extra chromosome on pair 21. Okay, that's instead of the 46 chromosomes we're supposed to have, that's 47 chromosomes, and it's called trisomy 21 because there are 3 on chromosome 21. Okay, so how does the risk happen for Down syndrome? Mother's age is one of the biggest factors affecting having the probability of having it. So if you are under the age of 35, you have a 1 in 1,500 chance of having a child with Down syndrome. If you are between the ages of 35 and 40, that number drops to 1 in 300 has a child with Down syndrome. And if you wait till the age of over 40, you have a 1 in 30 chance of developing or having a baby with Down syndrome. So that's definitely something that is related to age, because your chromosomes can't function properly to create what they need to, the right proportions of chromosomes. The next disorder that we're going to look at is Klinefelter's, and this is where you have non-disjunction occurring in your sex cells, so chromosome 23, and you have an abnormal sperm that unites with a normal egg. Alright, so by an abnormal sperm, we have an X and a Y instead of just one or the other, and your egg is just X. So you would have your 44 normal body chromosomes, autosomes. You would have 2 X chromosomes, one from mom, one from dad, and then dad would also give a Y chromosome as well, which will give you that 47 chromosomes. Sometimes you can even have 48 chromosomes if you have an extra X, so 2 extra X's. So instead of XY, you have XXY or you have XXXY, giving you 48. So because there's a Y chromosome, these people affected by Klinefelters are males. They are usually sterile, and they're usually tall with mild to severe retardation. Next, we have Turner syndrome. So this one is non-disjunction occurring in the sex cells again, so chromosome 23. Instead of having 2 X's, though, you have only 1 X chromosome. So instead of having 46, you'll have 45. So Klinefelter's is extra chromosomes, Turner's is less. So this one has 1 X chromosome for your sex chromosome. There is no Y. That means you will be female. And females with this disorder are generally short with underdeveloped ovaries, and that means they'll be sterile, and they will also have mild to severe retardation. And last, today we're going to look at karyotypes briefly. And karyotypes are basically organized profiles of a person's chromosomes in a picture that you can just clearly see and match up chromosomes. You pair them from 1 to 22, going from the biggest to the smallest, except for chromosome 23, that's going to always be at the end, and that's going to be your X chromosomes. So you might have 2 X's, you might have an X and a Y like our first example on the very first slide was, okay? And this is a quick way to tell if you've got a male or a female. So 2 X's here, this is a female karyotype. 2 X's here, this one's also a female, okay? So that's going to be it that we will cover for today. I would like you to be ready to do the next part next week on genetic engineering. If you have questions you need to ask in class, or come see me during pause, or send me an email, or find a way to get in touch with me. I am available during pause on Wednesday. I think we have pause from 1:30 to 2:30, so you can pop in anytime. If you need to be admitted to class or signed up, you have to request access. And then just let me know you've done that so I can go check, because it doesn't always let me know that I have people pending approval. Okay? Bring your questions to class. Have a great rest of your day. Bye, guys.